Hi, I'm LJ.
And I'm Elena.
Elena and I are students at the University of Minnesota Duluth and members of the American Fisheries Society. We like to keep up with aquatic sciences, especially with what's going on in the region.
In 2013, Large Lake Observatory researchers were granted funding to look at some changes happening in the western arm of Lake Superior. We talked to two of the scientists, Dr. Liz Miner, a chemist at UMD, and Dr. Bob Sterner, a UMD professor in biology and director of the Large Lakes Observatory.
We asked Dr. Miner what she was finding in the Lake Superior water samples she examined. Here's what she said.
The Great Lakes in general have not had a great deal of basic water quality and ecology work done on them. So we know that they're changing. We know that in the lower Great Lakes, quagga mussels have significantly impacted the ecosystem. They've removed the spring bloom of phytoplankton that used to exist. They've increased water clarity tremendously. Secchi depths are twice what they used to be in Lake Michigan. But we don't have a good feeling or a good baseline that we can look at these sorts of changes and project what's going to happen in the future relative to now.
When we asked Dr. Miner what her favorite scientific tool upon the Blue Heron research vessel was, she didn't hesitate with an answer.
My favorite scientific tool on the Blue Heron while we're out there is actually the CTD, because we get real-time measurements as the CTD, or conductivity temperature depth sensor, is going through the water column. It's measuring not just conductivity, temperature, and depth, but also chlorophyll, total suspended solids, the amount of sunlight, and other parameters that are of interest. And we get real-time projections of this onto screens in the lab as we're doing the sampling. And it's sort of like television for nerds. You say, oh, wait, where is the chlorophyll maximum now? Oh, that's cool. Wait, how much suspended sediment is there? Which kind of drives the technician a little bit crazy, because the screens are right by his chair and we're all clustered around staring to see exactly what the lake is going to show us. But it's a lot of fun, and it helps us to figure out where we're going to do our discrete water sampling.
A TV for aquatic science nerds. Cool!
Dr. Miner and other scientists participating in the project made many observations while they were on the Blue Heron. They had collected samples several times a year for 3 years from the same spots. These samples allowed them to measure nutrients, pH, particle abundances, and much more.
They also collected data using an autonomous underwater glider that collected measures of temperature, chlorophyll, and other pigments, and a moored profiling instrument that measures oxygen levels, currents, and water clarity.
What's cool about these unmanned technologies is that they provide a cost-effective way to measure more places and times than are possible with a research vessel.
The moored profilers can provide year-round observations like under-ice measurements that have never been made before in Lake Superior.
Some of the biological and chemical parts of the study are done in laboratories and include samples collected over the past decades to identify historic trends. The study produced the most complete and extensive data set of its kind. That's because of the gliders and profilers that collected a wide variety of data nearly around the calendar.
Because of the consistent sampling that's been going on, the researchers noticed a few things.
The thing that's gotten my attention most of all so far is a very strong connection between warm and cold years and the size of the phytoplankton, the size of the algae. So in a warm year, we typically find very small cells predominating. And in a cold year, we find much larger cells predominating. Now this potentially has really important effects on the food web, because different critters eat small cells than large cells. And so we're funneling food web energy into different parts of the food web depending on the overall lake temperature. The other thing is large cells tend to sink faster than small cells do. And so ecosystem composed of large cells is supporting different species, and also more of the energy that's being fixed by the phytoplankton in the lake waters ends up sinking out of the upper waters into the lower waters.
We've found a couple of things that are perplexing and interesting. One is that how acidic the lake is depends very much upon in-lake processes as well as out-of-lake processes. So it looks as if temperature and input of rivers affects things on a large scale, while within the lake between spring and summer, we see an imprint of the biology actually affecting the pH as well. And this is done because phytoplankton, the little plants in the water, take up carbon dioxide, and when they do that, they actually make the lake more alkaline. And bacteria that degrade organic matter actually release carbon dioxide and make the lake a little more acidic. So when you have the lake stratify in the summer, you have temperature changes that can affect the solubility of CO2, and then you have this imprint of where the eaters are relative to the makers of the food, and you get pH changes through the water column.
The research team also noticed that Cisco eat more native species like calanoid copepod than they did about 10 years ago, when the invasive spiny water flea composed of most of their diet.
Noticing things like changes in species composition and water chemistry may help us project what to expect if climate change trends continue into the future. Research like this also helps us understand other long-term changes.
Just for fun, we asked the scientists what it's like to do research and bob around on Lake Superior. Here's what they said.
My favorite sampling experience was a couple of years ago. We were doing sampling in the middle of the night, in the middle of the lake, and the northern lights were giving quite a show. So we were out on the deck. There were no running lights going on the deck because we were doing stuff that was light sensitive, and we had these amazing green spirals just rolling through the sky above us, and no anthropogenic light sources anywhere around except for us. It was really spectacular.
When the weather's good and it's daytime and you're out in the middle of the lake, it's a really special place. It's hard not to be just to look around and say, wow, this lake's amazing, and I'm privileged to be out here and working on it.
Large lakes have been shown to be critically important to our culture and to our life and to our enjoyment, and we need to be good stewards for the future. And part of being good stewards is understanding how they work so we don't break them.
We've still got a lot to learn about Lake Superior. It's what makes science exciting, and we're lucky to have people dedicated to studying the lake and people willing to fund such research.
Thanks for listening.
Stay curious.